The next abstract I was asked to comment on was the post hoc analysis of the combined ASCLEPIOS I and II trials, which compared subcutaneous ofatumumab with the oral formulation of teriflunomide in relapsing disease. And in this particular case, they were looking at NIDA-3. Just to remind you what NIDA-3 is no evidence of disease activity, which means no clinical attack, no new MRI lesion, and no particular EDSS progression, with a view to, I guess, trying to present ofatumumab as a superior medication to teriflunomide, which it was proven to be in the primary analysis of these two trials. So I don't think it was particularly surprising that when you do a post hoc analysis of a study that's already positive, the chances are you're going to get another positive result, and that's true for any kind of post hoc analysis. NIDA-3 is something that people are working towards as an outcome measure that is going to be standard today, but it has not been accepted necessarily by authorities. Hence the reason for putting it as a post hoc analysis, whereas most of these studies to get registered with the FDA would require a primary analysis usually based on relapse rate, which it had already proven in both studies individually and in the combined analysis. So at the end of the post hoc analysis, not surprising, ofatumumab showed a significant increase in NIDA-3 over the patients who were taking just the teriflunomide. Does this add anything to our knowledge regarding the efficacy of ofatumumab? I'm not sure. I think it's of interest. So what does this all mean with regards to NIDA as an outcome measure? Our problem here is we don't know what the natural NIDA would be. We know that in some placebo-controlled studies recently that the NIDA rate for even the placebo group was not insignificant, especially if there's not a lot of MRI studies done. The rule of thumb is the more you look, the more you find. So if you did an MRI every month, the chances of being NIDA at the end of a period of time would be almost zero, since MRI picks up a lot of different new lesions. But if only 1 or 2 MRIs are done, and maybe 1 or 2 clinical visits, so that there's not a lot of chance to pick up disease activity, you're going to see that even a placebo group would probably have a substantial NIDA rate. Buried in this study is that group of people who's by natural history are achieving NIDA. And we don't know that components in either the ofatumumab or teriflunomide group. Regardless, the ofatumumab patients more of those patients achieved the NIDA-3 at the end of the time period compared with the teriflunomide group, keeping in mind that there's probably a substantial portion of both of those who did not necessarily benefit from either treatment. I don't think that having these outcomes necessarily adds to my confidence that ofatumumab is superior than teriflunomide. I think the primary outcome told us that. It's just another way of looking at all the data compiled together. And I'm sure we're going to see other abstracts with subsequent post hoc analysis of the MRI studies and of some of the other metrics that were applied in this particular trial, which were not considered the primary or even secondary outcomes, but the tertiary and exploratory outcomes that one likes to achieve when you're dealing with a large study like this, to help eventually understand the population that might best benefit from a particular therapy. And although it's not proof that that population will benefit, it's the kind of hypothesis generating results that help guide us to either design new trials or to focus in and enrich a population that would be, say, the best responder to medication X over Y.